All right, y'all, welcome back to our next lecture on special senses. For this one, we are going to be going through our special senses, which includes our sense of smell, hearing, taste, and vision. So the sort of organization of the lecture is going to be, we're going to show you the receptors. We're going to talk about how the receptors transmit these signals into an action potential, and then we'll talk about how this signal is processed in our brain. So let's start with our sense of smell, right? So whenever we are entering new rooms or going into a new environment, we're always interacting with the environment, and one of the first things that we often experience is a new sense of smell in that area. And so, right, these are just things that could give a strong sense of smell, like in a new environment. But we detect smell and olfaction through our olfactory nerve, right? So one of our cranial nerves. And we have the olfactory tract and bulb, and then when we zoom into it, we see we have these olfactory sensory neurons that go into our nasal concha. So this area, right where the route of air is passing through, and air is going to have odorants in it, and there could be a lot of different odorants, and these receptors are able to pick up a lot of different odorants. And a lot of smells are going to be a combination of many different odorants. So it's really hard to say exactly which sort of neural pathway a specific odorant triggers. So we're just going to say, in general, odorants are triggering these olfactory sensory neurons. And so as you have molecules that are binding to these sensory neurons, they're going to trigger an action potential, right? So they're going to send action potentials onto then our olfactory nerve. And our olfactory nerve is going to relay this to our brain so that we can process it. So let's look at the mechanism of how these olfactory sensory neurons are actually picking up these smells. So if we have a generic odorant and it binds to its receptor on the olfactory sensory neurons, it actually works through something called a G protein coupled receptor. So this is called a GPCR. You may have seen this in previous biology classes, but this odorant binds to its receptor, and then it activates an intracellular protein called a G protein. And that G protein, once it's activated, and it gets activated with GTP, which is why it's called a G protein. It's like a protein that becomes activated when it interacts with this guanine nucleotide. So this G protein coupled receptor activates the intracellular G protein that moves down the membrane to a nearby enzyme called adenylate cyclase. And adenylate cyclase makes a specific messenger called cyclic AMP. And cyclic AMP is a really common second messenger that we see in biology. So the odorant would be the first messenger, cyclic AMP is the second messenger. And what cyclic AMP does is it opens a cyclic AMP gated cation channel nearby. So this would be like a ligand gated channel. So a cyclic nucleotide gated cation channel. Cyclic nucleotide gated cation channel, when it opens, lets in cations, such as sodium and calcium. And when that happens, this causes depolarization of these cells. So this is how these olfactory sensory neurons would depolarize in response to an odorant. And then, of course, this depolarization will be sent through our olfactory nerve and then to our brain for processing. So let's look at that processing step. If we have the odorants over here, the olfactory nerve is picking this up, the sensory nerves are picking this up and transmitting it to the olfactory nerve. Our sense of smell, so olfaction, is actually really interesting because it is the only sense. It is the only sense that doesn't go through our thalamus. So you might remember from our CNS lecture that the thalamus was sort of the master relay center for our sensory information, that when our body receives sensory information, it goes to the thalamus, and then the thalamus sort of tells this information which area it should go to, like the primary somatosensory cortex, the auditory cortex, the gustatory cortex, or the occipital cortex, right, for vision. But for olfaction, this one does not go through our thalamus. Instead, it goes straight to something called our limbic system, which is the amygdala and hippocampus primarily, and the limbic system. So these regions, we've talked about the hippocampus a little bit in terms of memory, and the amygdala is now a new structure that we're talking about. What this area functions in is in emotion and fear, and it's linked to memory. So that's why like certain smells can elicit either really strong emotions or very vivid memories. And we can also start to associate odorants with like fear as well. And so there's this like sort of emotional and like fear connection to creating memories that's tied to smell. And because this doesn't go through the thalamus and it goes through these structures instead, that's why we process odorants, or we can have certain memories and emotions tied to odorants that we don't necessarily have towards other types of senses. So our next one is taste. There's actually really interesting. There's a lot we don't know about exactly how our taste buds work, but on the slide has all of the different types of taste that we could detect. And these are just like some of my favorite foods, so I wanted to show it. But on our tongue, if we sort of zoom into regions of our tongue, this is sort of what like an individual taste bud looks like. And the taste bud has different taste receptors. So we have 5 types of taste receptors that we can detect. So 5 different types of taste. And those are going to be detected by what's called these gustatory epithelial cells. And these gustatory epithelial cells relay their information to then like primary sensory neurons. And you might remember from our cranial lecture that we have a couple of cranial nerves that function in taste. So we have like some that receive signals from certain parts of the tongue and some that receive signals from other parts of the tongue. But for this, we have our 5 different types of taste, and we have salty, sweet, sour, bitter, and umami. Umami is sort of the like savory flavor that comes from meat. So it's sort of a flavor associated with like rich, or like a rich savory flavor. So on the next slide, we're going to look at sort of how the like generic taste bud actually transmits this signal. So we have the taste bud over here, and then I've kind of zoomed into individual taste receptors. And I've pulled this image from another textbook because I really like this one compared to the ones we had in our textbook. So over here, we have sweet, umami, or bitter ligand. So these 3 types of taste function through a ligand-gated ion channel. And you see that ligand-gated ion channel is actually a G protein coupled receptor. So it's actually very similar to our olfactory sensation, right, that we started the lecture with. So when that G protein activates, it says signal transduction, but we know that GPCRs typically increase that second messenger called cyclic AMP. In this case, that cyclic AMP then opens up ion channels. In this case, they're calcium channels. So it opens up like intracellular calcium channels and surface level calcium channels. And that calcium, we kind of know from previous units that whenever calcium enters a cell, it triggers a release of a neurotransmitter. And that neurotransmitter for sweet, umami, and bitter tastes is actually called ATP. And we know ATP from previous bio classes. So ATP can function as a neurotransmitter as well. On the other side of this, we have sour. And we see that we haven't actually like identified the specific protein that sour is interacting with. But for sour, we are picking up acid. So hydrogen ions are what are coming through these taste bud cells. And then there's some sort of unelucidated pathway that hydrogen triggers in order to have an increase in intracellular calcium. Now, whether this hydrogen is activating maybe acid-sensitive proteins that then open up this channel for calcium, or maybe this hydrogen is directly causing a depolarization and opening up voltage-gated calcium channels. There's a lot of proposed mechanisms. At least as far as when this textbook was written, when I pulled this image from, it has not been quite figured out yet. For us, that's kind of nice, right? Because then there's less we need to know. So hydrogen comes in, something happens. Calcium comes in, triggers exocytosis. That should be very familiar, right? Exocytosis of some neurotransmitter. And then whether it's ATP or other neurotransmitter, then this is relayed through our primary gustatory neurons, where these neurons will send this information back to our brain. So let's look at how we perceive taste. Right? We have these primary gustatory neurons, which are going to relay this signal back to our cranial nerves that control taste, so our facial nerve and our glossopharyngeal nerve. And then there's also some slight taste functions, sort of like in the back of the mouth, of the vagus nerve. But these are going to send neurons back to our brain. So they're going to go through our brainstem up to the thalamus, and the thalamus to our gustatory cortex, which is in the insula. So we use 1, 2, 3 neurons in order to process this signal. And this is pretty typical, right? Because going through the thalamus, all of our sensation goes through the thalamus besides olfaction. So all of them will go through this pathway of primary sensory neuron, secondary sensory neuron to the thalamus, and then that final sensory neuron to whichever specific cortex it should go to. All right, the next one is going to be auditory. For auditory and visual coming up, there are a ton of structures within these systems. And by no means do we need to know all of the structures. I'm just going to highlight the important areas and talk about why they're important. So in general, for the ear, we have 3 parts of our ear. We have the external ear, we have the middle ear, and then we have the internal ear. And the external ear is going to be important for catching sound waves, right? So sound is going to come in, and our external ear will be able to funnel these and catch them and move them into the middle ear. The middle ear is really cool. We're going to actually highlight the middle ear quite a bit during this lecture. And if we zoom in here, we have the middle ear, right here. The middle ear has a few really important structures. The first one is called the tympanic membrane. You may have heard of this as the eardrum. This is what's going to be actually the first part that's catching sound waves. That's vibrating in response to sound waves. Connected to the tympanic membrane are 3 bones. We have the malleus, the incus, and the stapes. So these are 3 small bones, and they're often called the hammer, the anvil, and the stirrup. And these bones are really interesting because when the tympanic membrane vibrates in response to sound waves, the tympanic membrane will vibrate the malleus, and if it's acting like a hammer, then that one starts to sort of hammer on the anvil of the incus. And if the anvil starts vibrating because the malleus is hammering it, right, the anvil will start to vibrate and vibrate the stapes. So we have this transmission of vibration from sound from the tympanic membrane to the malleus to the incus to the stapes. And then this is going to interact with a really important structure in our inner ear called the cochlea. The cochlea is where we process, where we actually start to, to convert the sound information into ion movement and action potentials. So let's see how that happens. So what we're looking at, just to get oriented to the picture, we have the tympanic membrane, we have the malleus, incus, and stapes, and then we have the cochlea over here. So the cochlea, So the cochlea is a sort of tongue-like structure, and it's typically wrapped around to kind of look like a snail. So this picture is showing it sort of unraveling. It doesn't unravel like that, but this is just for simplicity. Okay, so going back to what we were talking about on the last slide, right? If sound waves come in it's going to start by vibrating the tympanic membrane, right? So vibrating the tympanic membrane. And that vibration is going to be transferred to those three bones, right? The malleus, incus, and stapes. Those three bones end up vibrating something called the oval window. And this is the opening to the cochlea. Opening to the cochlea. And the cochlea actually has like a fluid-filled space. So everything you're seeing in pink here is a fluid-filled space, and it does sort of have a directionality to it, where it's following sort of the arrow that I'm drawing. So if the oval window starts vibrating from the vibration of the bones, what that does is it actually starts vibrating the fluid, and it starts moving the fluid across this sort of inner membrane of the cochlea, which you can't see here, but I'm going to show you on the coming slides. Okay, so once the fluid-filled space of the cochlea starts to vibrate, we see here in step 4 that we have this fluid passing along what's called hair cells in the cochlea that I'm going to show you in the coming slides. And these hair cells are going to trigger the opening of ion channels that will cause neurotransmitter release of these sensory cells. So we have these hair cells, and as fluid passes by them, they bend to one side, and as they bend to one side, they trigger action potentials to be sent down, eventually our cochlear nerve. And so these hair cells have mechanically gated ion channels. That are bending a certain way in response to the movement of this fluid. So zooming in on the cochlea a bit, if we have that like fluid wave running over the cochlea has a couple key structures here. One of them is called the tectorial membrane. This is what's sort of like vibrating, and it's connected to the hair cells. Below the hair cells, we have something called the basilar membrane. This will be important a little bit later, but this is just like the basement membrane, right? Like that bottom structure that everything is sort of adhered to. But these hair cells are really interesting. Let me show you what they're doing in a little bit more detail. So we have 3 pictures here to show you what they can do. At first, they start off at rest. So if there's like no sound coming through, the hair cells aren't really bent to any one way. We see that there's still a very small amount of these ion channels open, and they produce like a tonic signal. And tonic just means like always on, right? You might remember that word tonic from our peripheral nervous system lecture with tonic receptors, meaning that they can always stay on a certain degree. So there's some like tonic level of firing of these. But let's say there's sound, and that fluid wave pushes across the surface of these hair cells. These hair cells start to bend to one side, and that bending opens up mechanically gated cation channels, and it depolarizes the cell. So we have these positive charges entering these sensory hair cells. It's eventually going to release or cause calcium to come in. So calcium will eventually come in and cause exocytosis of neurotransmitter, right? And then this would be like the primary sensory neuron that relays this information to the cochlear nerve, which eventually becomes part of the vestibulocochlear nerve, right? One of your cranial nerves. These hair cells can also be inhibited. So if fluid is pushing on the other side, then those hair cells will bend to the other side, and that closes the mechanically gated ion channels. So then we have no exocytosis of neurotransmitter. And so when we think about like how our brain is able to process like different pitches of sound, we actually do that with the structure of the basement membrane below, which is part of the cochlea, but below the hair cells. And so we see that we process frequency, so the pitch of sound, depending on like which region of that basilar membrane is actually vibrating in response to the fluid moving. So as that fluid is moving across, it's moving at a certain frequency, right? Because it's coming from a certain frequency of sound that vibrated the eardrum, the bones, the oval window. And so the frequency of this fluid moving is going to be matched to a certain region of the cochlea, certain region of the basilar membrane. And so this is like the unwound cochlea, right? We know it's usually circular, like spiral, but if we flattened it out, what we do is we process high frequency sounds near the beginning of the basement membrane or the basilar membrane, and then low frequency sounds near the end of the basilar membrane. And it has to do with the thickness of this basilar membrane and how it can respond to certain frequencies. So we are able to actually code for the pitch of sound based on where in the cochlea it's actually vibrating. For other things like loudness, that would be the amplitude of a sound, and that would cause more and more action potentials. So higher frequency of action potentials is coded by the amplitude of sound. But yeah, just for this, for pitch, it has to do with the region of the cochlea that's actually vibrating. Okay, so how do we process auditory information? So we have our one of our cranial nerves, right, our vestibulocochlear nerve, and it's getting input from the cochlea, but of course we have two ears, so the right and left cochlea. It's going to send these signals to our brain. There's actually some signals that go through our cerebellum because the vestibulocochlear nerve, yes, it's getting auditory information from the cochlea, but it's also getting vestibular information from the other part of the inner ear called our vestibular system. It's these fluid-filled semicircular canals, and the shift of the fluid in these canals actually helps orient our head in three-dimensional space so that we know sort of where our body is in space. So it's actually part of our processing of proprioception as well. But anyways, the auditory branch of this has to go through the brainstem, up the brain through the cerebellum, and then to the thalamus. Right? So the thalamus is going to be that relay center, and then the thalamus is going to send it to either the right or left auditory cortex. And what we see here too is that we do have some crossing over of sensory information, and we see this very often in processing sensory information, that we're able to process some sounds on our left side in our right cortex and some sounds on our right side in our left cortex. And this is really important too because it helps us, if we were to close our eyes and we hear sounds around us, we are able to tell where a sound is occurring relative to the position of our body, right? Even without seeing the origin of the sound. And that has to do with this sort of bilateral processing of information, and both the right and left ear individually send their information to the right and left auditory cortex. And then our brain is also able to code spatially for a sound because it's able to pick up on the time difference between when a sound hits your left ear versus your right ear. So if a sound is coming from a certain angle towards your head, it's going to hit your ears at slightly different times, and then our brain is able to process that and pinpoint the location of it, which I think is really interesting. Okay, last one, vision. This one is probably the most complicated one, but it's also really, really interesting. Similar to the ear, there's a ton of structures within the eye. We do not need to know them all. I'm just going to point out the most important ones. So we have the eye here. One really important one we're going to talk about is the lens, because this is what's going to help focus the light onto our retina. So our retina is going to be our portion of the eye that has photoreceptors. And this is where we actually transition from light signals to action potential signals. So the photoreceptors help interface with the external environment to transition that signal into an action potential. Right, similar to the cochlea, how the cochlea is what got the auditory signal and the vibrations, right, in a frequency of vibrations, and then it transmits that into a language our brain can understand, which is action potentials. So the retina functionally does the same thing, but of course it's picking up a different kind of stimulus here. It's picking up light with photoreceptors. All of the photoreceptors will then synapse onto neurons, and those neurons all come together to form the optic nerve, which is also one of our cranial nerves. Some other things here that are interesting that we'll sort of point out. The lens is able to be adjusted in terms of like how sort of squished it is or pulled it is, and that's because of muscles surrounding the eye. So we have like muscles here surrounding the eye that can pull this way, and we can like stretch the lens to help focus light. I do have a slide about the pupil because the pupil is going to help adjust how much light is going through our lens. And control of pupil constriction and dilation is actually an autonomic nervous system function. So pupil constriction is a parasympathetic function. You might remember this as being one of the functions of the vagus nerve, right? One of your cranial nerves, cranial nerve 10. And in order to test if the vagus nerve has been affected, you would shine a light in someone's eye. Because if you're exposing a ton of light into someone's eye, the eye wants to limit how much light is actually hitting the retina. So it's going to constrict the pupil in order to limit the amount of light going through. On the other hand, if you're in a dark room, this is going to activate your sympathetic nervous system, and its function on the pupil is for pupil dilation. This will allow a lot of light into the lens, and this makes sense in a low light environment that we dilate our pupils to let even more light in so we can try to focus our retina and see things in a dark environment. I have a brief reminder about receptive fields. It's important for our sensory system because we have different areas of our sensory system that are more or less sensitive. This is really important in the eye, and I'm going to show you in a little bit some optical illusions here to kind of hammer home this idea of large versus small receptive fields. Another thing I just wanted to remind you of is the ability of our sensory receptors to adapt, right? We were kind of talking about this earlier with tonic receptors, but sensory receptors can fall under tonic or phasic receptors. And a lot of the photoreceptors in our eye are going to be phasic receptors, which means they can't turn off. We do also have tonic receptors, though, in our retina, or kind of slower adapting receptors in our retina, because we still want to be able to like see things, right? But I'm going to also show you some optical illusions here that show you how we are able to turn receptors on and off, and that's that like phasic property of receptors. But before we get there, let's look at the retina in a little bit more detail, right? So up here we have a picture of light entering the eye, going through the lens, and the lens focuses this. By the way, the lens is a convex lens, and so as light is passing through this, we'll talk about it being convex a little bit more later. But eventually we're going to focus this in an area of the retina called the fovea. So the retina is this like whole back part of the eye, and the fovea is where we have what's called central vision. And central vision is where we have the highest visual acuity. That just means like the most clear vision. So if we were to look at what the fovea looks like in a little bit more detail, and what the retina looks like in a little bit more detail, we have two types of photoreceptors. We have rods and we have cones. Cones are for color vision, and rods are for black and white. So within the layout of the retina is that we have these photoreceptors in the back. So all of these over here, all of these are photoreceptors, either rods or cones. The cones are going to be the ones that look like they end in a cone. So the purple ones, and the rods are going to be sort of like the yellowish ones. But in front of these photoreceptors, we have the sensory neurons. So we have something called, in red here, we have bipolar cells. And then these are going to be ganglion cells. But these are those like primary sensory neurons. So these are going to be sensory neurons. Sensory neurons that relay this information to the optic nerve. So what's really interesting about our retina is that it's sort of designed backwards, because our photoreceptors are in the back of the eye, which means if light is like trying to reach those photoreceptors, it kind of has a hard time because it has to go through all of these layers of cells in front of it. So the bipolar cells, and the ganglion cells. So light has a much harder time reaching the photoreceptors in our retina on our like peripheral vision versus our central vision. So our fovea here, our fovea is our central vision where we have the highest visual acuity, which is really just to say we can only see clearly exactly where we're looking. I know that sounds kind of weird, right? But if you were to like look around your environment, wherever your eyes are fixated on, that's your central vision. Everything in your peripheral vision, so on the sides, you can still see, but you can't make out a lot of detail, right? And part of the reason for that is because of this orientation of how the retina is built, how we have these neural cells in front of the photoreceptors, so light can't hit these photoreceptors in our peripheral vision as well as it can hit the photoreceptors in our central vision. The other concept with this, though, is that rods, generally we have rods on the peripheral side of our, let's just write this down. So rods are generally in our peripheral vision. And we typically have cones in our central vision. And this sort of makes sense, right? Because rods are just for like black and white, just like kind of is there light there or not? And in our peripheral vision, we don't need really high visual acuity, right? Because the purpose of our peripheral vision is just to see like if anything is coming in or out of our visual field. And evolutionarily, right, this would just be like, is there a threat or is there movement that could be a threat? So as long as our peripheral vision is able to detect a movement, that's actually good enough for evolution. It's really our central vision where wherever we're currently looking, that is where we have a really high visual acuity, so we can determine really fine details with our central vision that we can't do with our peripheral vision. And then the cones that are concentrated in our fovea, we have three kinds of cones. We have blue, red, and green cones, and we're able to activate a combination of these in order to sort of create different colors. And these cones are spanning sort of major wavelengths within the visual spectrum. Okay, so I have a sort of series of optical illusions here, and this is going to hopefully help hammer home the idea of small versus large receptive fields and tonic and phasic receptors. So for this one, I want you to stare at the plus sign. I'm going to put some images on some videos on the side, but I don't want you to shift your focus to the videos that I'm putting on the side. I want you to just continue staring at that black plus sign. So start staring at the plus sign. So you probably see some videos on the side of some animals running, but don't look at them. Keep staring at the black plus sign. Okay, now I have a question for you, right? Not that you actually are like submitting this anywhere, right? But this is just for you to think about. Which animal was running on the bottom video? Was it a lion, dog, cheetah, or squirrel? So if you want to try to pause and think about it, you can. But what this question is showing is our peripheral vision, right? Our peripheral vision being sort of bad at determining fine details. So we couldn't tell maybe exactly what was running on the bottom video. You probably saw two videos of something running, but you don't know exactly what was running in each. But that's okay, right? Because the point was like, could we detect motion in our peripheral vision? It's just like, is light there and is something moving? And this shows that we have a difference in receptive fields in our fovea versus our peripheral vision. So within our peripheral vision, we will have very large receptive fields. Right? So peripheral vision has large receptive fields, which means we can't tell a ton of detail. If you're wondering, it was a lion on the bottom video. If you would like to go back to confirm, there's a lion and it's a pug on the top video. When I was first building the side, I didn't realize that in the top video, the pug like sort of runs over like a baby pug. Yeah, it's kind of funny. But yeah, this shows the difference in the size of receptive fields in our central vision versus our peripheral vision. I have another sort of optical illusion here for you. We have all of these gray lines, and for this one, I want you to actually like look around the grid. As you look around the grid, you might see some black dots. And as you start to move your vision, it sort of seems like these black dots are sort of appearing and reappearing, right? And if you take a second to just like fixate on one black dot, the black dots around it start to sort of disappear. And then if you move your eyes, other black dots start to disappear. So you really are only looking at the black dots where your central vision is. And every time you move to a new black dot, the ones around them start to disappear. And it's just because of our peripheral vision not being very good at picking up details on the sides. This is, though, also getting into the idea of these receptors can sort of fatigue or they can adapt, and we start to ignore certain stimulus depending on the color gradient. So I'm going to show you a couple more. So the previous two that I just showed you were to really kind of get the idea of small versus large receptive fields, that we have small receptive fields in our fovea and large receptive fields on our periphery. The next two are all going to be about our central vision, and it's really going to be about cone adaptations. So the tonic versus phasic. So I'm about to put a picture on the screen. I want you to stare at the black dot and don't look away. And try not to blink a ton, at least in the beginning, but if you do need to blink a few times throughout it, you can. Okay, so I want you to stare at the black plus sign. So keep staring at the black plus sign. You probably see some purple dots in your peripheral vision, making a heart. But as you continue to stare at the black plus sign, those purple dots might be going away. Like you might kind of see them coming in and out. But keep staring at that black plus sign. Okay, I'm about to transition it to a white slide, but keep looking where that black plus sign is. You should see green dots now where the purple once was. And if you kind of move your eyes around, you see that green heart sort of following where your central vision is. And it's probably just about gone away by now. So really interesting, right? Like the purple was in your periphery was fatiguing cones in that area. So cones that were able to detect purple, right? So like a combination of cones. And when cones that are picking up that color start to fatigue, then when we see white, and right, of course, white light is like all wavelengths of light in the visible spectrum, but the cones that pick up or can detect that certain color of purple are now fatigued, so they sort of drop off. And then what is left is the opposite, so green. So that's why when we transitioned here, we fatigued the sort of like purple sensing photoreceptors. And when we saw white light gave all of our photoreceptors all forms of light, right, all wavelengths of light, then we only saw green instead because we had fatigued, right? So this shows like phasic receptors, that our cones have a phasic property that they can sort of turn off when they're exposed to a stimulus for too long. I'm going to show you one more. This one's really interesting. Same idea, you're just going to stare at the black dot in the center of the screen. And this video is actually on a timer, so it's naturally just going to shift. All right, so stare at the black dot. When the colors change, I want you to continue staring at the black dot. Okay, so keep looking at the black dot. Don't move your eyes. Okay, so like right now you probably see like orange sky, purple trees, blue grass, but keep staring at the black dot. Now when it changes, I want you to actually move your vision around. And you probably see the picture is actually in black and white. But when you look back at the black dot, it goes back to like green, blue sky, green grass, green tree. Let's do it one more time. Okay, move your eyes around and then return to the black dot and everything should be in color. But then move around, it's black and white. So if you wanted to go through these a few times, you can. But it's all showing the same thing, that these cones are able to adapt. And it's the same idea. It's like when we fatigue cones that absorb a certain wavelength, and then when we give sort of like more white light, where it's receiving all wavelengths of light, only the cones that are not fatigued are now firing, and that gives us that like inverted color to where it looks like everything is actually in color. but it's actually in black and white, but we have just like sort of like tricked our eyes and our photoreceptors into like turning certain ones on and off. Okay, so that was fun, but let's do the physiology now, right? Like, how do our eyes actually convert light or dark into action potentials? So when we are in the dark, when there's no light, we have no action potentials. Let me show you how that happens. It works through a mechanism called dark current. So what we're about to do is called phototransduction, and we're going to do it in a rod. The mechanism is the exact same in in a cone, but we're just going to do it in a rod because it's a little bit simpler. Okay, so when there's dark, so no light, we have two channels here. We have one called a cyclic GMP gated cation channel. So actually very similar to a previous cation channel we've looked at. And then potassium leak channels. These potassium leak channels are always, just letting a little bit of potassium out. And as potassium exits, that causes hyperpolarization. You remember from our neuro unit that leak channels, they only let a little bit of ions through. They don't let a ton, so it's a very weak sort of hyperpolarization. Okay, so how do these two channels interact and cause no action potential to occur? And of course, when we're referring to no action potential, we mean on the sensory neurons. So it's going to be on these like ganglion cells over here in blue, right? Because those are the sensory neurons. Okay, so when it's dark, there's a high amount of cyclic GMP present in the cell. I'm going to show you a slide a little bit later about how it actually gets there, but you don't need to know how cyclic GMP gets there, just that in dark, there is a high amount of cyclic GMP. That molecule will go and open the cyclic GMP gated cation channel, and then we will have a big influx of cations, so calcium and sodium. So this causes depolarization. That depolarization is going to travel down the photoreceptor. Depolarization travels down the photoreceptor, where it will open up voltage-gated calcium channels, sort of on the terminal end of the photoreceptor. Very similar to previous neurophys units, right? When calcium comes in, it triggers exocytosis of neurotransmitter. And then we have this neurotransmitter released onto the sensory neurons. Okay, but here's the thing. This neurotransmitter that it releases is an inhibitory neurotransmitter. So this would be something like GABA. GABA is a really common inhibitory neurotransmitter. And when GABA stimulates the next cell, it typically hyperpolarizes. So this is a hyperpolarizing neurotransmitter, and it lets something like chloride into the cell. So it causes it to go further from threshold. So the idea here is if we have a photoreceptor here, right, this is cell 1, we have that bipolar cell 2, and then we have that sensory receptor 3. Let me change colors here to maybe black. In dark, there is a high action potential frequency on the photoreceptors, but because they release an inhibitory neurotransmitter, we have low action potential frequency of bipolar cells, which translates to low action potential frequencies in the sensory neurons. So then we perceive this as like there is no light information, right? So no action potentials to the brain, or less action potentials to the brain. Okay, let's do the same mechanism, but this time in light, and it's really just the reverse, right? So when there's no light, when there is light, we have a low amount of cyclic GMP, which means this channel does not open. Potassium continues to leak out, so the cell hyperpolarizes. So it slows down its release of action potential, which means these voltage-gated calcium channels aren't opening as frequently, and we significantly reduce neurotransmitter release onto bipolar cells and sensory neurons. But this is good, right? Because this neurotransmitter that we saw on the last slide is inhibitory. So if we reduce the inhibition, we actually cause these next neurons to then start to fire. So again, if we have that 1, 2, 3, here, we would reduce the action potential frequency of the photoreceptors, but that increases the action potential frequency of the ganglion cells and the bipolar cells. This sends light information to our brain, where we then process it as action potentials. There is light information here. So it's a little bit confusing, right? How we have this sort of inhibition on our photoreceptors, and it sort of reverses the signal. And this is what gives us no action potentials or less action potentials. Oh, sorry, this is supposed to say convert light into action potentials. So not the word no there. This is how we convert dark into less action potentials, and then our brain doesn't process it. And then this is how we turn light into action potentials, and our brain does process it. This is the fancy textbook diagram of phototransduction. The mechanism we just did is called phototransduction. We see there's a few more proteins here, but this would be the light scenario, and this would be the dark scenario. So you see this is dark current because in the dark there is current of these ions causing release of that inhibitory neurotransmitter, whereas in light we stop that neurotransmitter release or lessen it. But just as a reminder, right, this is, we are sending action potentials to brain in this scenario. And in this scenario, we are not sending action potentials to brain. That's because of the fact that this neurotransmitter released by photoreceptors is inhibitory. If you would like to, you could look at some of these other proteins here that are actually causing cyclic GMP to be either high or low. It's really similar to the previous steps we did, though, right? We have a G protein, and we will either activate or deactivate that G protein in order to cause cyclic GMP to be high or not. So you don't need to know it in this detail, but if you would like to, here's the fancy textbook picture. Okay, just a couple more slides on the visual system. So going back to the fact that our lens is a convex lens, typically when we shine light through convex lens, it actually inverts the image onto whatever surface its light is passing through, right? So this picture here is showing that this image is inverted onto the other surface once this light passes through the convex lens. And this is actually what happens with our retina. Because our lens is convex, the image that's projected onto our retina is actually inverted, so it's upside down. But of course, we perceive the world right side up. So it's a really interesting combination of sort of how our brain is processing this information. Our brain is taking into account a lot of different things in our environment in order to perceive the world as right side up. And it's actually an interplay of multiple systems that is doing this processing for the brain. So neural information from the visual system is going to be processed in our visual cortex, but also we were just talking about earlier that we have something called the vestibular system, right? And it's part of our inner ear. And there's going to be sort of crosstalk between our visual system and our vestibular system that can tell our brain that our body is upright and the world that we have in front of us is also upright. So our brain takes into account sort of the position of our head, the position of our vestibular system relative to the visual information we're getting. And we sort of, for a sort of non-scientific word, right, we sort of fix the image in our brain. But it's really just multiple neurons taking into account position of your head, position of the object, and our brain is able to understand that the world we're interacting with is actually right side up. But it's really interesting, right? Because what a convex lens does in terms of light coming through it, it actually reverses the image. Okay, so the last thing is just the processing of it, just like we've done for every other system. So we have our left and right eye here, and we see that both eyes contribute to our visual fields. We have a right visual field and we have a left visual field, and then we have overlap between them. So what's interesting about this, right, is that our right eye contributes to our right visual field and part of our left visual field. And similarly with our left eye, right, it contributes to the left visual field, but also part of the right visual field. And then we have this binocular zone where the visual fields are overlapping. So the retina is going to process this light information, send it through the optic nerve. The optic nerve does crossover at an important structure called the optic chiasm. And then we have crossing over where we process information from our right visual field and our left visual cortex. And then we process information from our left visual field and our right visual cortex. So similarly to other crossing over events we have in the nervous system, this is what gives us more orientation within three-dimensional space, and this is what helps give us binocular vision. The fact that that we can process both visual fields sort of in both cortices in the back, and that crossing over helps us perceive that. Okay, that's the end of special senses. It's a little bit longer of a lecture, but I think it was really interesting. I hope you enjoyed learning about the special senses.